like from starting to thank organizers for opportunity to share with you our progress on this very interesting project. And it's basically focused on the Golgi complex, which is a very fascinating organelle. And it has a unique architecture of stacked cistern and can be divided in several subcompartments. The first subcompartment of a Golgi, this compartment or entrance side of a stack, is involved in reception of secretory material from endoplasmic reticulum. Next compartment, the co-Golgi region or medial Golgi, consists of stacked cistern, where a number of protein and lipid modifications occurs. And finally, trans-Golgi network or trans-Golgi compartment usually participates in sorting of protein and lipids to their final post-Golgi destinations. This is how Golgi look like in electron microscopy, and this is pretty close to what you can see in the textbooks, the stack of cisternae. However, in light microscopy, the appearance is slightly different, and it looks like a pretty compact perinuclear organelle. So during their life, cells need some Golgi sometimes to be generated, and this happens in a number of physiological situations, like cell cycle, when more Golgi should be inherited by daughter cells, or increased secretory load when Golgi has to handle a lot of secretory material arriving and moving through these organelles. And finally, in cell differentiation, when such cells as neurons or muscle cells have to generate additional Golgi units. So the main question of our project is how transcription contributes to Golgi biogenesis. However, this question was kind of neglected by Golgi field and was really poorly understood. The other problem it was difficult to study transcriptional responses related to Golgi in context of physiological situations because they were contaminated by other upregulation of genes like mitotic genes in cell cycle or cell-specific during differentiation. And another problem was a lack of a robust experimental model for de novo Golgi biogenesis, which could be used for transcriptomic studies. So to set up such system, we engineered a HeLa cell line expressing mannose 2 HRP. So these Golgi-located enzymes with HRP tag allow us to deposit within the Golgi a rigid polymer of diaminobenzidine. In this way, we can kill the Golgi, the old one, and force the cell to generate the new Golgi and observe how it evolves in time. Here you can see a movie where we deactivated the old Golgi with diaminobenzidine. It looked black. And in green, you will see generation of the new Golgi in cells where the old one was killed. It happens in the timeline of several hours, and in electron microscopy is represented by several studies where we can see old Golgi to be dispatched and sequestered into lysosomes, while new Golgi elements form, and first they look pretty ugly, not well organized. However, by 10 hours after inactivation of an old Golgi, cells can make very good stack with bona fide architecture. And at this point, we started to look also at the molecular composition of newly forming Golgi and found that it also differs during different time points. For example, at 6 hours after inactivation of the old Golgi, we start to see appearance only of some marker here shown in green. Then by 10 hours, we start to observe almost full set of Golgi markers in some cells, meaning that this represents a particular time point in the biogenesis of Golgi stacks. If we look at the entire timeline of the organelle generation, we can say that first detectable Golgi elements start to be seen at 2 hours after inactivation of the old organelle. Then cell generate what we call ugly Golgi, whose polarity and trafficking activity is still poor. And then by 10 hours, almost perfectly organized Golgi emerges, and it's also fully functionally capable. So to understand how cell transcriptome changes during this process of Golgi biogenesis, we collected the cells from each time point and then subjected them to single-cell RNA-seq. Why we used single-cell RNA-seq? Because of heterogeneity of Golgi recovery phenotypes. This field in the picture represents the typical appearance of the cell at 10 hours after Golgi inactivation. And here we have really different phenotypes. So judging on a couple of markers, we can say that in some cells, they appear both in new Golgi, indicating a complete recovery. In some cells, only one marker appears, so this is partial recovery. We also observed no recovery at all. We observed dead cells. And finally, in some cells, the level of monosidase to HRP expression was not sufficient to inactivate Golgi. So we were doing nothing, basically. At this point, we obtained RNA-seq data, and here its outcome. So this UMAP shows only cells expressing monosidase to HRP. And each cell, each dot represents a single cell transcriptome, and the cells were clustered based on similarity of their transcriptomes. So when we started to look at the clusters time point by time point, we observed that at the beginning, from 0 to 6 hours, clusters emerge along the time points. And only at 10 hours, we start to see more clusters which persisted over the time. And this probably indicates the different cell fates. To understand these fates, we made gene enrichment ontology analysis. Gene ontology enrichment analysis for modulated gene in each cluster. And we found that in cluster 2 and in cluster 10, number of activated Golgi transcripts. So to prove this Golgi gene activation further, we looked at the expression. Here you can see in the map the cells which express the highest level of this Golgi gene UST, and it perfectly overlaps with cluster 2. However, at this point you can ask me probably the question, how do you know that these cells basically make the new Golgi? To prove this point, we made fish. So we looked at the transcripts of Golgi genes during the process of Golgi biogenesis. So what we found that at time 0, when we just inactivated Golgi, and it can be seen here in black, the number of USTM transcript was pretty low, while in the cells recovering the Golgi, the new Golgi, and it can be seen here in green, the amount of UST mRNA strongly increased. We observed this phenomenon also for other Golgi genes, so they were upregulated and exhibited higher mRNA levels, as also confirmed by quantification. So at this point, we started to ask questions. First of all, we wanted to understand transcription of which Golgi components is activated during de novo biogenesis. Different functional classes of Golgi genes, or different compartments, or some activated, some no. And the other question which we addressed was, how does transcription activation correlates with Golgi morphogenesis, and also recovery of its trafficking function. So in terms of compartments, we observed that a lot of trans-Golgi genes were activated. However, also cis and medial Golgi genes were well represented. The same can be said for different functional classes. So genes encoding protein or lipid-modifying enzymes represented a big chunk of activated gene, as well as structural and trafficking genes, together with a number of other functional groups. But the most important and interesting finding was when we looked at the dynamics of Golgi gene activation and how it correlates with Golgi morphogenesis. If you look at this heat map of more than 100 activated Golgi genes in our set, you can probably see that this heat map divided in two parts, and there is a clear switch in activation between 6 and 10 hours. So I would like to remind that this 10-hour point corresponds to full recovery of the structure and function of the Golgi, and it seems that transcription strongly contributes to this process. So we wanted to understand whether this dynamics of Golgi gene transactivation is similar for different compartments, and this 6 to 10 hour switch was typical for cis, medial, and trans-Golgi genes. Moreover, the same observation we did for genes encoding different functional classes of Golgi protein. Here I show the example for glycosylation enzymes and coat and adapter proteins. So at this point we can say that massive activation of the Golgi genes coincide in time with the recovery of a stack architecture and functional reactivation of a Golgi organelle. And that induction of Golgi genes occurs in a pretty orchestrated manner and does not depend from the compartmentalization of the gene product or their function. So at this point, obvious question to address was which transcription factor drives this process? And we started to study it. And the other question was how does the transcriptional program works in the physiological context? So for transcription factor, we made a specific analysis using bioinformatic tool, TransFac predictive software. And that's used the input of the Golgi genes, which we observed during our transcriptomic study. And TransFac does following. It just identifies common transcription factor binding site in gene promoters. Here, probably you can see these green balls, which are common transcription factor binding sites. And on the basis of this analysis, it predicts these transcription factors. This prediction returned the list of several candidates together with number of target Golgi genes. However, at this point, we also wanted to look at the expression in HeLa cells, and we found that some were expressed really poorly, indicating that they should be discarded from analysis. So we ended with a curated list of transcription factors and silenced them. And finally, we observed how the silencing affects Golgi recovery process using inactivation tool. Now, these slides show control. When we looked at the cell field here, you can observe in green Golgi apparatus in untreated cells. We deactivated Golgi by diaminobenzidine and observed efficient recovery in the presence of control mRNA. So when we silenced the transcription factors, we found that some of them didn't have any impact, while suppression of others seriously impaired recovery of the Golgi complex. And this was true for CREB3L4, for MITF, and also for COOX1. And finally, we looked also of impact of silencing at the electron microscopy level. And here we observed striking result. For example, MITF silencing cells never recovered good Golgi observed in control cells, indicating that indeed this transcription factor could be involved in gene activation process. So at this point, I would like to summarize this part, saying that MITF, CREB3L, and COOX1 represents a good candidate's transcription factor for biogenesis. And we also can be pretty sure that MITF suppression blocks the transition from an immature to mature Golgi. With this, I would like to wrap up my talk and acknowledge my partners for the project, Alexei from the States, Jan from Netherlands, and members of the lab. The project was carried on by Francesco Forno and Domenico Abete in my lab. Here you can see them. And I would like to thank particularly Bioinformatics Core of our institute, and especially Diego Di Bernardo for many helpful suggestions, internal and external collaborators, and you for your attention. Thank you.
Questions?
My question is, if you've looked at, within your transcriptomics to see if the peroxide-killed Golgi somehow triggers the new Golgi process within that 6 to 10 hour time frame, and if you've tried, and this follow-up to that is, have you tried other ways of getting rid of the old Golgi, such as laser ablation, and do you see similar transition times?
Yeah, we tried different thing to kill a pre-existing Golgi, and it's really tough, okay? So this is the only way actually to kill it, because we tried also to ablate using laser to cut away part of the cell. But the throughput was really, really low, okay? So you can have maybe 2 cells out of 20, okay? And when among these 2, 1 die, so you can't collect very precisely. The other problem was that there are ways to kill, pre-existing Golgi, like with Brefeldin A, okay? So this drug would dissolve Golgi completely. However, in this case, the problem is all components of all Golgi are floating inside the cell, okay? So when you wash out BFA, they just come immediately together. But previously, in the studies of group of Rainer Peppercock, we found that the timing was more or less the same, because they cut away using the laser Golgi from the cell, and the recovery was more or less within the time which we observed, indicating that it's required transcription and generation of new Golgi components. So what triggers, we think that it might be unfolded protein response, okay? Because at a certain point, all all cargos go outside of the Golgi and carry chaperones, and then probably they are not returning back, okay, chaperones, because Golgi cannot make trafficking in a sufficient way. This would lead to accumulation of new proteins that would trigger. So we are studying this now, okay.
Thank you.
I was wondering if you tried repeat exposures for the diaminobenzidine, if you're treated again at like 18 hours when the Golgi components are already upregulated, is it faster?
Yeah, no, we didn't do that. It would be interesting, let's say, exercise, okay? And what would you expect? More activation, let's say, or faster generation, okay? Yeah, faster generation, it could be a way, okay? Although it's a little bit stressful for the cells when you make diaminobenzidine inside the Golgi, but it could be a way to accelerate de novo formation. So you kill the new Golgi, okay, and expect that the newest will be generated faster, right? Maybe it may happen.
So, nice talk. I'm just trying to appreciate your model a bit more, and that's why this question. So you say you kill Golgi and you are expressing this enzyme, and then when you get in, what you get in return by your transcriptomics data, alteration in, again, Golgi-related enzymes, either involved in glycosylation and so on and so forth. So is there a kind of a feedback loop which is actually deriving this biogenesis, and that would be obviously energetically and otherwise favorable for the cell, and which feeds back, and that's why, I mean, you get the Golgi, keep getting it back after this arrest and so on and so forth.
Yeah, we think that it might be a regulatory loop, okay? The problem is to find a sensor for this loop, okay? Because our biggest question is how the cell senses the absence of a Golgi, okay? And there could be different mechanisms. One could be related to the impaired glycosylation, which somehow can be sensed without glycosylation enzymes. And another could be, you know, lack of structure of Golgi protein. So the cell starts to understand that there is no Golgi structure which is functional anymore.
So if I may, very quickly, I mean, so I agree with your problems with the Briffelden A experiment. It can be very nasty and so on. So let us say, I mean, if you break this loop away from the Golgi, which is, let us say, in the trafficking pathway, maybe some RAB enzymes and so on and so forth, and you block the trafficking, which obviously is critical for Golgi biogenesis, and go back and see if you get similar results, because then you are actually not fiddling around with the Golgi per se directly, but a pathway which is involved in Golgi biogenesis.
Yeah, this could be the case, and also scenario to investigate. Yeah, very good. Yeah, nice idea.
Okay, thank you again.